So, good day to everyone that who is taking part in this particular webinar. I would like to introduce Professor Benali. Professor Benali earned a state engineering degree in process engineering and MSc in medical physics, and now in the final step of PhD in medical physics from University of Science and Technology, Hawaii, Biomedicine. For the education and employment record, since January 2014, he is working as a permanent professor in Ekahid Hamma Lagdar University. LOAD for the teaching activity. He is teaching general physics, biophysics, and scientific writing, scientific English in Faculty of Nature and Life Sciences. Concern the scientific production, he have many publications in different journals such as European Physical Journal, European Journal of Medical Physics, and Journal of Instrumentation. For the communication, he participated in different international conferences such as IMIC 2013, TESANAT 2015, ECMP 2016, ICRA 2017, SIPM 2018, and SNTMA 2018, and ECMP 2018. Concern competencies for data processing, his programming languages Fortran and C++, and mastered the simulation Monte Carlo with code MCNP. For experimental techniques, he has experience in control tools in medical physics such as characterization of the scanographic installations and calibration of the dosimeters and radiation beams in radiotherapy. Coming to his research interests, he is interested in fields of radiation protection, oncology, radiation therapy, applied physics, medical physics, biophysics and healthcare technologies, computational biophysics, and Monte Carlo simulation. He is a editorial board member for the Journal of Endocrinology and Exocrinology for known publications, and his current webinar will be covering an overview on bioinformatics. Professor Benali, you may just go ahead with your speech.
Okay, thank you, Mr. Pavan, for this introduction.
Thank you.
Thank you. Welcome, everybody. We hope you are well and healthy in this difficult period with COVID-19. First, I would like to thank Mr. Vijay, Genome Publication Director, and their team for inviting me to be a speaker on their webinars. And I hope this webinar will be successful for you and add new. So I will share now my screen. I would like to share my screen now, okay? Are you see the PowerPoint now?
Yes, it is visible. It is visible.
Okay. The outline of presentation will be introduction to bioinformatics, databases, and sequence alignment, and primary databases, and we will finish our presentation to last update on COVID-19 using the popular resource on the NCBI. Introduction. We'll start with history of bioinformatics. Original of bioinformatics. The study about uptake of pure DNA from an infection bacterial strain could confer virulence to non-virulence infectious strain by McCarthy in 1944. Then the role of DNA was validated by Hershey and Char in 1952. And Sanger and Thompson published in the latest 1950s the first sequences for amino acid of protein. From 1958 to 1962, the first bioinformatician, Dijkhoff and Leidy, was developed the comp protein software encoded with Fortran language. And Dijkhoff also published in 1965 the first atlas of protein sequence and structure. The first published multiply sequence alignment algorithm was a generalization of the Needleman-Winch algorithm, which involved using a scoring matrix in 1970. Then the first DNA sequencing method to be widely adopted was the Maxam-Gilbert sequencing method. The first software dedicated to analysis Sanger sequence was published by Roger Staden in 1979. Then a model of evolutionary change in protein by Dijkhoff et al. Published in the Atlas of Protein Sequence and Selector in 1978. In 1987, the manipulating DNA with polymerase chain reaction known as PCR, which allowed to amplify DNA without cloning procedures. The genetics computer group at the University of Wisconsin published the eponymous GCG software suite in 1984. This was the first software collection developed for sequence analysis. Another sequence manipulation suite was developed after GZC in the same year, known as DNA Start software, which could be run on PC. In the years between 84 to 85, other sequence manipulation suites were developed to run on Apple II and Macintosh computer. The most importantly during this period that MBL and GenBank and DDBG sequence databases have unified in the years between 86 to 87 in order to standardize data formatting. And today, this union still exists and is now represented by ENSDC. In the early 1990s, the Berners-Lee at the European Organization of Nuclear Research initiated the World Wide Web. The MBL was made available online with their databases such as SwissProt and Rebases in 1993. Also, in 1992, the GenBank databases became the responsibility of the NCBI before it was under the Los Alamos National Laboratory. Then the NCBI in 1997 was made available online, including the BLAST tool, followed by genome databases in 95 and PubMed in 97 and the human genome in 99. The first complete genome sequencing of a free living organism was sequenced in 1995. The second generation, also called next generation sequencing, NGS, that started with 454 Piro sequencing technology. This technology allowed sequencing 1,000 to million of DNA molecule in a single machine run. Since 2008, the arrival of massively parallel sequencing technology. This resulted in an exponential increase of sequencing in public databases and new respiratory infrastructure arose for model organisms. The emergence of research specialized in this field of bioinformatics, there is a significant increase in user-friendly tool often available throughout the interactive web server like Galaxy and helping community such as SecAnswer and BioStar. There is also an explosive need for bioinformaticians on the help job market in academic, private, and government sector. The International Society for Computational Biology published guidelines and recommendations of core computational that a bioinformatician should have a curriculum for using, applying, and analysis. This is a good paper. If you need more information, I suggest for you this paper, Brief History of Bioinformatics. Definition: Bioinformatics is an interdisciplinary field that develops method and software tools for understanding biological data, in particular when the data set are large and complex. As an interdisciplinary field of science, bioinformatics combine biology, computer science, information engineering, mathematics, and statistics to analyze and interpret the biological data. Bioinformatics has been used for in silico analysis of biological theory using mathematical and statistical technique. Now, how can biologists use bioinformatics? There are three proposed ways. The first way is to start with lab data, followed by computer analysis. The both study can give us new knowledge. The second way is to start with lab data, followed by computer analysis. Then hypothesis to compare between the both study to know the reason of similarity or difference. Then we come back to lab for validation. The third way is started with public data, then followed by computer analysis, then to obtain new knowledge. The impact of bioinformatics on biological research. Gene-based drug discovery and drug designing, disease diagnosis, identification and characterization of genes, discover functional relationship among organisms, and discover genetics relationship of different organisms. The challenge of bioinformatics. Biological data are not started in one database. Also, the different databases have different rule and style patterns. Variability of data annotation. Also, the different solution for the same problem, the need for keep us with the data. Where we can get into the games, there are typically there are 3 stages of genomic and bioinformatics project. The first stage is data generation, followed by data management and data analysis. The data generation stage have the genomics and all the omics data. Then for the second stage, we need data curation, data storage, and data presentation. The first stage need data meaning, inferring similarity, and pattern prediction. A global opportunity of bioinformatics. It does not require expensive resource. It just need an idea, data, personal computer, and a good internet connection. Also, accessibility to the most databases is free. Now, databases. This is a typical gene bank. In the past, biologists should follow the sequence line by line to determine the start codon, etc., etc. This worth long and large time. This is a paper published in 1984 in the Journal of Biological Chemistry. This is location of gene, and this is sequence. Here we pose a question: can we search in this paper? Can we modify or update? Can we cross-reference, or can we use the sequences for further analysis? The response is it's very, very difficult to do search or update or cross-reference and probably impossible to do to use the sequence for sequence for for the alignment, for example. What is databases? A database is a collection of information that is organized so that is be easily accessed, managed, and updated. Databases can be classified according to the type of content: bibliographic, full text, numeric, and image. Where we can find the databases, the most biodatabases are typically hosted by servers that are accessible throughout the internet. This is an example from the protein databases and UniProt databases. This is the NCBI homepage. We selected the protein databases and we do a search about severe acute respiratory syndrome coronavirus. As you can see here, all fields, so the SARS, COVID-2, and COVID-19. The search gives us 11,747 result. This is the UniProt databases. Search in these databases with the same keyword, we found 41 reviewed paper and 456 reviewed. This is the GenBank pharma of the coronavirus. As you can see, a lot of information about what we are looking for. For example, this is the GenBank have 7,096 acido amino. It updated in 30 March 2020. This is a journal total, etc., etc. There are a lot of information provided by the databases. This is the future, and this is the origin, which represented the sequences of this of the COVID-19. Data organization and presentation. Experimental data are submitted directly into the databases by a researcher, and the data are accessed essentially archival in nature. Once give databases accessed number, the data is primary databases are never changed. The databases should be clean and reliable data, comprehensive and updated annotation, and use-friendly searching and trivial. Primary and secondary databases. Research around the world or submit their data raw to primary databases. There are three primary databases: is NCBI, EBI, and DDBG, followed by data curation, computationally or manual, then obtained to secondary databases and arrive to and is available now to for the user. Information retrieval from biological data. There are three types: text search or sequence alignment, for example, with using the BLAST tool, and browser genome data, as we found it in NCBI. How we should store data file. This should require standard formatting to avoid repetition and confusion between sequences. This is DNA sequences, and this is protein sequence. FASTA format. The FASTA format is text-based format for representing either nucleotide sequence or amino acid. A sequence in FASTA format begin with a single line description followed by a line of sequence data. The description line defined in distinguish from the sequence data by the greater than symbol at the beginning. It's recommended that all line of text be shorter than 80 characters in length. Black line are not allowed in the middle of the FASTA input. FASTA, the accepted for nucleotide and acidoamino. The sequences are accepted to be represented in the standard format amino acid and nucleic acid code. The nucleic acid code supported are we have A, T, C, G, and U for RNA. This is the N for any, and this is G or T, A or C. This is G or T or C, etc. And this for the amino acid. Sequence without annotated are useless. But why? As you can see here, the annotation is very, very important. For example, the code, the barcode or the ID number, it's like the accession number, and the set annotation, it's like the information in the card, in the ID card, and this is the sequence. Typical gene bank. There are three principal parts: the header, started by the locus, and the footer, then the sequence. If you need detail of the gene bank type, you can see in this link. The PDB text format protein structure. This is an example. The PDB format provides standard representation for macromolecular structure data derived from X-ray diffraction and an MRI study. This is the detail of each part of the PDB text format. If you need more information, see this link. The PDB 3D format. This is the protein data bank, is a data base for the three-dimension structural data of a large biological molecule, such as protein and nucleic acid. The data are freely accessible on the internet via the website of its member organization, PDB, PDBG, and RCSB, and the BMRB. The PDB is overseen by an organization called the Worldwide Protein Data Bank, WWPDB. And this is the PDB is a K in area of structural biological, such as the structural genomics. Most major scientific journal now and some funding agency now require scientists to submit their structure data to the PDB. Sequence alignment. What's sequence alignment definition? Sequence alignment is a way of arranging the sequence of DNA, RNA, or protein to identify a region of similarity that may be a consequence of functional, structural, or evolutionary relationship between the sequences. Sequence alignment can be between two sequence pairways or between more two sequence as multiply sequence. Sequence alignment has two forma, global or local alignment. This is an example. Let's align this to sequence. So the alignment to identify the match and mismatch and using the gaps. There are score. Each alignment have a score. Score is function of match, mismatch, and the gap. Comparison between global and local alignment. This table gives us a comparison between us. This is the local alignment. The local alignment do the alignment in the local region with the highest level of similarity. As you can see, the high level of similarity, and it does not assume over the entire length. Contrary for the global alignment, align across the entire length of the sequences, and assume similarity over the entire length of the two sequences. The global alignment, it's good when the two sequences are closely related, and so global alignment approach may give false result if the two sequences are not closely related. Primary databases. There are three primary databases: NCBI, EMBI, and DDBG. The main integrated databases or tools. More recently, the databases have shared and integrated information from a variety of databases to produce complex, comprehensive site with access to information in most databases from a single site, and in some case, an integrated databases. Three of the main integrated databases in use today include the entry for the NCBI and the sequence retrieval system for the EBI, and DBG ET and link DB for the DDBG. Great strength of these databases is that they not only allow you to search them, but they provide link and handy pointer to additional information in related databases. The three systems differ in their databases and the link they made the information. This is the homepage of the European Molecular Laboratory and the European Bioinformatics Institute, EMBL, EBI. The European Bioinformatics Institute is an international government organization which are part of the European Molecular Biology Laboratory family, focus on research and service in bioinformatics. It's located on the Wellcome Genome campus in Hinxton near the Cambridge. This is the tool data resource provided by the ABI. There are important tools and important databases. This is the UniProt page. The Universal Protein Resource, or UniProt, is a comprehensive resource for protein sequence and annotation data. The UniProt databases are the UniProt knowledge base, UniProt KB, the UniProt reference cluster UniRef, and the UniProt archive UniPark. The sequence retrieval system, SRS, is a data integration platform that provides fast access to diverse type of biological information regardless of the data format. The APIs SRS serve provide access to a large number of publicity available biological databases. SRSB databases. The type of databases include, for example, are sequence and sequence-related metabolic pathway, etc. SRS programming. The core of SRS consists of the library of function to perform, query, and extract and parse entire for flood the data bank in their original format. This is, if you need more information, I suggest for you this paper, or visit the link services at the API homepage. The DNA Data Bank of Japan. The DDBG Center collect nucleotide sequence as a member of ENSDC, the International Nucleotide Sequence Databases Collaboration, and provide freely available nucleotide sequence data and supercomputer system to support research activities in life science. It's located at the National Institute of Genetics, NEG, in the Shizuoka Prefecture of Japan. The DDBG exchanges its data with European Molecular Biology Laboratory, EBI, and with the NCBI. There are a lot of good partners for DDBG. For example, the Database Center for Life Science, DBCL, was established in April 2007. And the National Institute of Genetics. Also, the Protein Data Bank of Japan and the GBG, Japan Alliance for Bioscience Information. The DBGET and link. DBGET is a sample database retrieval system for a diverse range of molecular biological data set. Database is considered simply as a set entire which may be stored in a single file or multiple file. This is few information about DBGET. Generally, the DBGET has a simpler but more limited search method than either SRS or entry. SRS of API and entry of the NCBI. The second principal databases, is the LinkDB. It's databases of link, as you can see in this picture. The link contain all cross-reference link, original and reverse link, extracted from all the databases in DBGET. Thus, the link in the LinkDB are as the following three topics: original, reverse, and equivalent. Now, see National Center for Biotechnology Information. The NCBI is a division of the National Library of Medicine at the US. It's located in Bethesda, Maryland, and was founded in 1988. The NCBI study computational approach and provide online delivery of biomedical information and bioinformatics tools. The NCBI hosts approximately 40 online literature and molecular biology databases, including PubMed, PubMed Center, and GenBank, that serve millions of users around the world. This is the homepage of the NCBI. This is the resource, and this is the welcome, and this is the popular resource. The popular is not, it's sorted by the most used, and this is the support center included get starting, resource, popular, future, and NCBI information. The entry databases is a molecular biology database system that provide integrated access to nucleotide and protein sequences data, gen-centered and genomic mapping, information, 3D selector data, PubMed, Midline, and more. The system is produced by NCBI and is available via the internet. Entry cover over 39 databases, including the complete protein sequence data from Peer International, PRF, SwissProt, and PDB, and nucleotide sequence data from GenBank that include information from AMBL and DDBG. The entry retrieval system use an initiative use interface for rapidly searching sequence and biography data. A unique feature of the system is its use of pre-compute similarity search for each record to create link to neighbor. This is a paper if you need more information. Now we will see the latest update on COVID-19 using the popular resource of the NCBI. We will start with the homepage of NCBI. This is the homepage of NCBI. And this is the popular resource sorted by the most used and not in alphabetic order. We start with PubMed, then Bookchat, etc. We will do research with the keyword COVID-19 in each one of these databases or tools. Let's start now with PubMed databases. PubMed databases, this is the old interface of the PubMed, and this is the new one. PubMed comprise more than 30 million citations for biomedical literature from Medline, Life Science Journal, and online books. Citation may include link to full text content from PubMed Center and published website. This is a 3D article and latest literature. And this what about online? It's related with Midline, PubMed Central, and Bookchat. So after you can access directly to the PubMed and write your keyword. For COVID-19, we find 5,340 wait, as you can see here, we have 5,348 about COVID-19. You can in the display option you can selected best match, sort by, selected by publication, etc. For example, we selected this paper. This paper is full text link. For example, this literature on COVID from the PubMed. It's very easy databases, etc. There are a lot of literature in these databases. The second database is Bookshed database. This is the interface of this database. Here there are the new update. I selected two books, this one and this one, and I tried to do COVID-19. I found just two books published. This is the first one. It's published in January 2020. It's available online for online reading, not PDF format. The second book, according to COVID-19, is this is one published in March 2020. This is other book from the latest update. And this is one. The PMC. The PMC. This is the interface of PMC. The public PubMed Central is free full text archive of biomedical and life science journal literature at the USA. National Institute of Health's National Library of Medicine. There are more than 6.1 million articles, full participant, etc. This is our research about COVID-19 at PMC. We found 3,741 results. Selected the first one, you will see. This is the first one, severe acute respiratory syndrome, Corona 2, SARS-CoV-2 and coronavirus disease 2019. Citation for PubMed article make up the second largest component of PubMed databases. You can download from a PDF directly from this link. This is the nucleotide databases. This is the research about COVID-19 in this database. I will do the research about COVID-19 and SARS and SARS and coronavirus. The first one is COVID-19, and this is the GenBank format. This is accession number, and this is the footer, and this is their origin sequence. The second one is this is the graphic format. When you move the mouse, you can sort a lot of information. This is the third format, is FASTA format. For the SARS, severe acute respiratory syndrome, you will find this result accession number. We need this result to do BLAST or to do pairwise sequence alignment later. This is the bad coronavirus. Also for protein database, This is the result. We have 11,766 result about COVID-19 in protein database. This is the link of the Center of Disease for Control and Prevention in the US. This is the GenBank pharma of SARS-CoV. The basic local alignment. The basic local alignment. The basic local alignment search tool find the region of local similarity between sequence. The program compare nucleotide or protein sequence to sequence databases and calculate the statistical significance of match. If you would like to do sequence alignment with the tool BLAST, you can put the accession number in this first box. Then this is the home page of the BLAST local alignment. You can select it, nucleotide BLAST or protein BLAST. This is an example of the using of BLAST. We can put accession number in this box, then check all parameter. This is sequence alignment with all databases at the GenBank. It's multiply sequence alignment. You can select it, show add a new result, a new windows, and click at BLAST. This is this is the result of our BLAST with COVID-19. The name is BLAST COVID complete genome. There are three principal statistical value: the carry, cover, the e-value, and the pair percentage of identification. If we do this is pairwise multiply sequence, you can click it align two or more sequence. You can put the first, the carry, for example, it's the COVID-19, and this is the SARS, and we do click with BLAST, you will obtain it. This is the result of the alignment. We have 82.3 percentage as percentage of identification. There are the carry cover 88%. The second one is BLAST COVID with bad coronavirus. We found 70.56 percentage as percentage identification, and just 3% of the carry cover. The third BLAST is with bad SARS. We found 81.66 as a percentage of identification, and the carry cover 87 percentage. It's appeared now that this is the best sequence alignment. So the best is with the SARS. If you need more information about the BLAST, you can see in this link which BLAST program should use. Depending on the nature of the sequence, it's possible to use different BLAST program for the database search. The most commonly used method is to BLAST a nucleotide sequence against a nucleotide datablast or protein sequences. BLAST-N is in fact a rather poor tools for finding protein coding sequences. The obvious solution to this problem is to use BLAST-P rather than BLAST-N whenever possible. If only nucleotide sequence are available, plus X plus N or plus or T plus X should be used, provided we are searching for protein coding sequences. Also, another good reason for translating the query sequence before the search is that you get protein hit which are likely to be annotated. This is the GT generic test registry. The genome databases you can access databases. Human Genome Project research completely sequences the first human chromosome and deposit the sequence data at NCBI. This is the PeopSham database. It's good one for especially for the chemistry researcher. We do research with COVID-19 in this database. We founded this result. There are 115. There are a lot of information in this database. For example, we selected the chloroquine. This is the chloroquine. There are a lot of information here. For example, molecular weight, etc. There, the structural. This is for the chemical and physical property, and this is for experimental property of the chloroquine. Spectral information and literature, for example, Spring Nature reference, etc. For the genome and map resource, you can access in the genome data viewer human genome with a lot of way. The first way is to go to the resource and select the genome viewer, or go directly to genome map and genome data viewer. Then you can write which organism you would like. For example, we selected human. This is the human genome resource at NCBI, and we selected. You can select one of this chromosome, and you can download also. This is when I tried to download it. There are for the blast for the FASTA format. There are it's 920 megabyte, and this is the third chromosome of human genome. I selected this number 3. This is the chromosome number 3 of human genome. A lot of information. SMP databases contain human single nucleotide variety, micro satellite and small scale, etc. Getting start, submission, access data. For example, we do search about SARS or COVID. We don't find any result in this database. For the gene database, integrated information from a wide range of species. Recent may include nomenclature, reference sequences, map, pathway, etc. The link for the using gen, the gen tools, and the other resource. This is our research about COVID and or SARS-CoV-2. We found 32 results. How to submit if you are a biologist or researcher and you find the new sequence, how you can submit your result or your sequence into the CPI. You can follow these steps. You can go directly to the submit. This is to the submit. Then you pop up this page, and you can select my submission. It's required. This step required sign up to NCBI. If you click to my submission, you can then this page can selected one start new submission. For example, I selected Jambank. This is the new pop up this page, and click to the new submission. Then followed, for example, I selected influenza virus. As you can see, the name is not required. You can provide a title to help you identify. Second one is to submit. Fortunately, I haven't sequenced, so I can't continue this. I will do directly delete the submission. Don't be lie. If you need more information, I suggest for you the NCBI minute how to submit your 6S RNA data to submit in this YouTube video. International Nucleotide Sequence Data Collaboration. This is the home page of the NC, the NC-NAS-DC. It's a long-standing foundation initiative that operated between DDBG, EMB, EBI, and NCBI. It's covered the spectrum of data, raw reads, throughout alignment, and assembly to functional annotation enriched with contextual information relating to the sample and experimental configuration. If you need more information, you can see in this paper, International Nucleotide Sequence Data Collaboration. This is the nucleotide acid research databases summary. It's a good one. This journal, the first issue of each year, is devoted to bibliographic databases, and an issue in July is devoted to paper describing web-based software resource of value to the biological community. This is the new version of the paper, the issue of 2020. There are a lot of related journal, NAR Genome and Bioinformatics, and NAR Cancer, etc. If you need more information, you can see in this link. This is a few URL and place to learn more. The lecture of Dr. Yaqub Ashab or Sabi M. Tami, etc. If you have question, or you can send me at my emails.
Uh, Professor Benali, are you done with your presentation?
Yes.
That's great. What do you see? Like the field of bioinformatics will be growing in future, or it's going to be stagnant? Because there are many queries from upcoming students stating that how come the bioinformatics career would be, and choosing the bioinformatics where they'll be led.
I suggest for the student to don't use a lot of. You can selected one, for example, to as a beginner student, you can selected just one databases, for example, NCBI, and one lecturer or professor go step by step. Then we can, after that, after we have the basic background about the bioinformatics, then we can go through to the other databases. I suggest the NCBI the databases as first databases for the beginner student.
Okay, that's great.
Yeah.
That was a very good presentation, Professor Benali. Like you have explained very very in detail about different type of databases and how we have to search and how the databases would be and all. Yes, I personally have some knowledge on this type of databases. Probably like that NCBI, PDB, DD, BJ, and all. Yeah. So, what about the current research on bioinformatics level? Like, what is the level of bioinformatics research on all over the globe?
At where? At Alwad University?
Yes, about your
Uh,
university.
yes. Currently in Algeria, we haven't possibility to do sequencing, so it's very hard. Just personal work at Algeria at all, not just in Alwad. It's contrary for India, for example, Turkey, Egypt. There are the good opportunity to do research and in the bioinformatics.
Oh,
We
that's
haven't
great.
center, not yet have center in Algeria, not yet.
Okay, okay.
Bioinformatics.
Fine. So, what about your interest to publish something on this bioinformatics research and all in the terms of your current research interests? So, can you merge your current research interests with the bioinformatics and can you publish something?
Of course, currently I am not yet starting as a medical physicist. I focus in the medical physics, and now as a medical physicist, I interested in the cancerology,
Hmm.
and this way we can relate it between medical physics and bioinformatics in the cancerology. Currently, I am supervised few student in master degree in this, for example, for breast cancer and colorectal cancer, etc. Bioinformatics, it's good tools to know more about the cancer. This is my interest in the future.
Okay. Okay,
Yes.
that's great. So you have also mentioned about local and global alignment in the database search and alignments of sequences. So are we still using Smith and Waterman algorithm for the local alignment, or are there any other algorithms that can be used for?
For the needle is for the global alignment,
Yes.
and for the local alignment, it's Waterman.
Yeah, Smith and Waterman. So the same
Yes.
algorithms we are making use of, right?
It's uh, I don't know exactly what you would like of
Ah,
question, but
yeah, my
of it's
question.
dynamic. It's dynamic sequence alignment. But the inconvenience of local and global alignment of global and the dynamic alignment with Needleman-Winch or with it's the time worth with this two with this both method. It's
Okay.
contrary for the blast. It's very quick tools.
Okay.
So blast is preferred than the local and global with Needleman, Winch, or Witterman as a local alignment.
Okay. And what is the best way to have a multiple sequence alignment? For
What's
multiple
best tool?
sequence alignment, yeah, best tool. I actually know about cluster W. Is there any other tool that can be used for that?
Yes, it's I showed in the presentation about the blast. It's the best is the protein P, the
Oh,
blast P. Now
okay,
it's the
okay,
best one. Yes. If you need about just specific the gene, you can use the blast N. Yes.
okay, okay. That's a very good session, Dr. Benali. Like we and our participants have actually had a lot of information gathered from your valuable presentation, and yeah, thanks a lot for your time. And if you are done with your presentation, we'll just close the session.
Yes. Yes. Thank you so much. You are welcome.
Yeah, thanks
In
a
the
lot,
next
and I would,
next
yeah,
webinar.
please.
Thank you all. Thank
Yeah,
you
I
for
would like
the
to.
genome. I would like to transmit all my thanks to Genome Publication, and I hope all successful of your journal.
Thank you. That is possible only with cooperation and guidance of eminent people like you. So I request you to be, active in the participation of the journal, and yeah, we'll have to cope up with the submissions and get the journal indexed as soon as possible. I wish your hands to be always open for us, and yeah, we'll feel very free to contact you anyways.
Okay, thank you, doctor. Thank you,
Yeah, I
doctor.
would like to.
Thank you so much.
Yes, sure. Thank you. I would like to thank and deliver my vote of thanks to Professor Abdel-Hai Benali for his webinar topic and delivering a valuable speech to all the participants. And I would also like to thank all the participants to take part in this webinar. And yes, this webinar will be published online in both YouTube and also in our websites. You can go through the webinar sessions anyway anytime you wanted to. And yeah, in this particular COVID outbreak, everyone please be safe, and we'll be with our research team like Dr. Benali and other persons and on our editorial board members. We will always get back to you with the updates on COVID-19 then and there. Thanks a lot, and I'm now ending the session. Thanks a lot.
Okay, thank you. You are welcome. Thank you so much.
Thank you.
Thank you so much.